Hello and welcome to this Eppendorf custom webinar where experts share their knowledge and vision for their work in bioprocessing. Today we are joined by Diogo Perez dos Santos, bioprocessing engineer at the National Institute for Bioprocess Research and Training, which is based in Dublin, Ireland. Diego will tell us about his work on how modeling can help to achieve more precise bioprocessing approaches. If you have any questions to Diego after watching this webinar recording, reach out to him or to us. The contact information will be available at the end of the talk. And now, without further ado, I hand over to Diego for his presentation.
So hello, as Stefan said, my name is Diogo. I'm a bioprocessing engineer here at NIBRT. So I'm gonna show you a bit about our research, how I'm doing precise bioprocessing through modeling here at NIBRT. So first, I would like just to showcase that my name is Diogo, so a bit different because it's a Brazilian name, but that's how it's pronounced. Stefan did great. So now a bit about myself. So as you know, I'm a bachelor's engineer with a master's degree and PhD in chemical engineering. So I was working at the Brazilian animal pharmaceutical company here at the south part of Brazil. So right down in the red dot. And then what happened? I crossed the Atlantic and came here to Ireland. So how did it happen? What happened for me to get here? So I was recruited by Professor Sax Mantalaris and Nick Panascaltzis that they have a grant about metabolism-driven precision manufacturing of cellular therapeutics. That's great thing for Research Ireland. So the logo is right down there. So they need a bioprocess engineer to develop this approach of modeling in their grant. So now that you know who I am and what I'm doing here, how I got here to Ireland. So let's talk a bit about our work. So this grant is about an interdisciplinary approach to address heterogeneity by understanding directing metabolic changes in the cells. So there is composed of four main pieces. That's one is bioinformatics, that's gonna construct a metabolism database. One is cell biology that's going to characterize this heterogeneity by single cell analysis or population analysis. Then there is bioprocess engineer that's going to increase precision through modeling, using models to help with that. And then there's also analytics that's going to define metabolic biomarkers. So all these areas work together to make a interdisciplinary approach to tackle this challenge of directing heterogeneity that's happening in all the cellular therapeutics products nowadays. So, and what's my task? My task here is being the bioprocess engineer. I'm working with the task about bioprocess engineering. So I'm trying to increase precision through modeling, making models for these cell types. So what did I do? I do develop these models. And the mathematical model is just equations that can predict cell behavior. And then we use it to optimize two main factors. That's one is the culture media. So think about the food that the cells needs, the nutrients that they need to grow and characterize and be quality to make their therapeutic activity. And then the feeding schedule as well. So you need to think when and how much of that nutrition you need to give the cells in the right amount in the right time, so the cells not overfeeded or even are not starving as well. So everything we do is to think about the critical quality attributes, as you know, that things that make the cells have their therapeutic potential, reach a therapeutic dose, and then do the activities that we know they're capable of. So everything we do here is to think about metabolism, because even the best cells are not going to thrive in an environment that doesn't feed them enough. So metabolism is the key to understand what they need and how they need it and how it's going to control their fate in the bioprocess. So, and how I'm doing this modeling. So basically here I'm going to show you a basic process. So if I have a bioprocess, I have a bioreactor here. Normally I'm going to be one of the main things we do monitor is the viable cell number or concentration and the cell number or concentration. That's a key process variable in my bioreactor. And in empirically, we know that they are controlled by the growth rate. So how fast the cells can grow is going to determine how many cells we have in the bioreactor, and the death rate is going to control how many dead cells are going to have as well. So they are controlled by this parameter that I created. And we know that this parameter is dependent on some nutrients. So think about growth rates is going to be dependent on glucose. That's the main source of energy for the cells. And the death rate is going to depend on some kind of toxic metabolite. That's lactate. So like if the lactate is super high, then death rate is going to happen. It's going to increase, and then that cells are going to start appearing in a bioprocess. But then we can have a different effect as well, so a negative effect. So if you think that the glucose is high, then it's going to affect negatively the death rate. And if lactate concentration is low, it's going to be good for the growth rate. So then you're going to start making connections between the nutrients and then the rates that you have, the parameters that you have. So we're going to be building that map that's going to control everything. And these dots, that the connecting dots, are going to be equations. So I'm going to explain what's the equation that's going to control that, and then the simulation can try to calculate it and then reach the actual growth rate, actual death rate, and then reach actual viable cell number and that cell number. So this is just a simple model. So in the final model, you're going to have several more nutrients and like all the amino acids, more toxic metabolites like ammonia and other things. So this map is going to be way more complicated, but that's the basic model, so you can understand what we're doing. So then, if I know everything, so I'm going to first digitalize my system. So I'm going to put my bioreactor and my feedings that I'm going to use. Gonna try to control how long it's gonna happen. Then here's the image from the GPROM software. So what I start to do, I start to write these equations. They're gonna be adding more equations to the system. So to explain every metabolite, to explain every growth parameter I have. So everything is gonna be inserted in the model in the end. So in a final model can have like hundreds of equations. So some of them are going to be normal equations, some of them going to be mass balance and differential equations. So everything can be done here. And then after I have everything set up, so I can run the simulation. But now you're thinking, the cells are complex systems that can be really hard to understand. We have been doing for centuries and still don't understand everything that cell can do and how it do. So how can a bunch of equations, so the simulation, be realistic and not just assumptions or a kind of hallucination? So how we can trust that the equation is going to be a real predicting cell number and other things? So what we're going to do, we're going to use experimental data. So we use real data, real experimental data to train this model. So after being trained, and then we can calculate some kind of statistical significance. So we can check if the model is really predicting cell behavior. So that's how we know that the model is good. And then once we prove that the cell is realistic, we can use it. Now we're going to have a digital copy of our cell that can be run as many times as possible. So it's really useful. But now, you know that I'm going to need the experimental data. So how do I do it? So for us, we choose the DOSBox as our platform because it's a parallel bioreactor, multi-parallel, so we can run a lot of bioreactors at the same time, and we can monitor all the common variables that we're going to have, the parameters, pH, O2, temperature, CO2, and we can even put feeding, so flow of media going into the system and a removal pump as well, so we can take media out of the system. So we choose that because we can now run multiple experiments, because we can incorporate feeding and perfusion, so making the bioprocess optimized as possible. So we're gonna use tissue flask as well to start, but then we want to use a high throughput system so we can generate data faster, and then we can really optimize their process and be really scalable process as well. So that's why we chose it. So that's why we are using. And then what kind of data are gonna be needing for this DASBox run? So I'm gonna need to understand the cellular heterogeneity. So that's why the cell biology is gonna be really important. So I'm gonna try to understand the metabolic signature. So what's gonna be driving cells to be different? I'm gonna be needing some kind of data about cell composition, because normally there are some cells that differentiate or become are modified in some way. So I need to know what's going to be the effect of that modification as well. And basically, I'm going to need to know the growth kinetics as well. So I'm going to need to understand how fast they grow, how much glucose they consume, how much amino acid they consume. So everything I'm going to need to know by the whole population, some subsets, because then you can have some specific part of the cells that can be more consuming than the whole population. So I'm going to need to try to understand that as well. And metabolism in general, but thinking here about as well the energy metabolism, because energy is the main thing. So if the cells cannot generate energy, then they are gonna die. So is really important for us. So ATP generation. So that's basically the data that we're gonna need. And now gonna see how we can develop this model. So the pipeline is a blend of experimental modeling. So things it's in vitro and things are in silico. So then we're gonna use this approach. Being some days we're gonna be in the lab preparing reactors, and some days gonna be sitting in the computer running simulations. So first we can start with the model development using a software like GPRONS, make the model, and then we then need to go to the lab to do the first reactor, the first bioprocess run, so we can get the initial parameter values, calculate everything, so we can start using that values in the model. Then we can test the model, see if the structure is correct, if it's showing growth of cells, and then that phase, a growth phase, so everything as we expected and we have seen in the lab data as well. Then we can run what we call a sensitivity analysis that's gonna try to make a statistical relevance of every parameter. So it's gonna then say to me that one parameter was really a key factor in determining the cell growth. So then I'm gonna need to go back to the experiments to really be sure that I capture this right number. So then we can go back to the lab to make more experimental runs and then optimize it. And after going that, I can do even more parameter estimation, try to correct the predictions, train the model with the data that we now have, and then be trying to finalize it. Then, as usual, we go back to a lab, do more experimental runs. Those ones are not going to be used to train the model, just they're going to be used to verify the model, to make a validation analysis. So it's going to try to use the data, the parameters that was generated from other runs to try to predict that run. And then I can do some calculations, and then it's going to say that the growth is validated. That's going to be predicting correctly viable cell number, that cell number, and the other parameters that I find useful for us. That's how we get to a validated model. So, but now I think maybe you understand what I'm doing, but do you understand why do I do it? Why do I need to have a validated model? Why I would want a digital copy of a cell How can I use it? So that's the most interesting part. So I'm gonna show you a real use case scenario that was made by my group, but a few years back, that was using a mouse myeloma cells that produce a specific antibody that detects colorectal cancer. So what they did back then. So first you have the experimental data, that's the blue square. So you see a cell that's growing over time, and then normally it reaches maximum value and then starts to die. And then we have this experimental data. Now we can try to make our model. So try to do the development pipeline, try to construct everything and train the model. And then we get this line, this dotted line. So now we have the simulated data as well. So then we have a simulation that's not perfect, but capture the train line. So it was growing, then stopped growing. So then we have now think about that it's now a validated model that's a digital copy of our cells. So what we're gonna do with that? Now we can start arguing instead of going back to the laboratory and test new experiments like start feeding some new nutrients to the cells. We can just instead of going to the lab, I'm gonna ask the model. So what can I do to make these cells grow more so stay healthy and reach a quality attribute that I have now? So I can start testing a lot of scenarios, and then I can even ask them. So try to maximize the viable cell number before the toxin metabolite accumulates as well. So then I can get this red dotted line. So it is the answer of the model that says if you start feeding, you're gonna be able to grow the cells into this data point, but then they're gonna die eventually because of another toxin metabolite. So we have now, I think that was did not cost to run, so it's just a simulation. So now it's saying to me that there is a way to optimize it. So then what I need to do is to go back to the lab and test this theory that the simulation that your digital copy said that was possible. So that's how we got the red circles as well. So then you see that applying the feeding strategies that the model said that would be good, I could do what he predicted. So I could grow the cells for longer than expected, and even a bit more than the model predicted, but with a close estimation. And then we did optimize it with just a few runs. So normally these things, a lot of experimentation, a lot of trial errors, and so it takes a long time. But using modeling, we can do it faster and easier than the traditional approach of trial and error. So that you see, if you have a digital copy of your system, you can optimize it in a way that's going to be way less cost intensive. So that's why we do digital copy of ourselves. And then we can even have different approaches of how to optimize it. And we can even link the experimental data with our modeling. And then the modeling is gonna even try to correct our bioprocess run in real time if we can. So if you connect the model with the real physical system. So that's what can be done. But that was done in the past. So what are we doing now? So now our current focus in cell therapy. So we are using 5 cell types are really interesting, uh, being really deeply researched in this area because they all have like a really huge impact in cell therapy. So I think cells that have been heard about, like CAR-T, MSCs, or iPSCs. So cells that are being used in cell therapy, but they all need some kind of optimization, some kind of understanding how their metabolism works. So then what we want to do is to be able to use this approach of modeling so we can scale up this production, make the cells grow more faster in a lower time, a shorter time frame of development, because they already be using, but they are all really costly. And then if they stay in this price, they are not going to be broadly available for the population. So that's our driving motive. And then if you can do this, then you're gonna drive down the treatment cost, because their cells gonna be able to reach cell number faster and a higher cell number, with keeping their quality as well, so the therapeutic potential. So that's all. That's our goal. So that's and then that's why the group is focusing now. So that's what I'm doing. I started with NK cells, but I'm gonna be eventually doing all the cell types. So that's it. I hope you understand now how we can use modeling to optimize a bioprocess to help with the development of a bioprocess. I hope you like the presentation. And then if you do bioprocess well, think if you could do a simple modeling as well to try to predict it. So prediction is a really useful tool to be prepared to when you need to stop a bioprocess, when you need to go and continue longer. So I think modeling is getting really, I think, more easy to approach than it was back then. So now they have a lot of softwares as well, and coding is getting simpler as well. So I think if you think about it, think if you can apply it as well in your bioprocess. So, and if you want to know more about our research, so check our website, thecell.ie, and then follow us in LinkedIn and X as well. So I hope you see the fruits of our labor in soon enough. So thank you and have a nice day, guys.
Thank you, Diogo. I think I like the presentation very much, so I can answer your question already there. Thank you for this very insightful talk. And thank you, the audience, also for watching the recording. And as said, if you have any questions, reach out to us or Diogo. For more information about Eppendorf Bioprocess, you can also visit our website via the displayed link. Here you can also see again the email where you can contact us. We want to thank you for your interest and hope to see you soon in the next presentation. Again, thanks, Giro, for your nice talk. Goodbye and have a good day, everyone.